Basically what WormBase does is to take what you output and then organize the information and then give it back to you. Now the problem is there are a lot of U's. So we want to make sure U1, whatever U1 is saying, is understood the same, the same meaning by U2. So that's what basically ontologies help us to do that. And so today I'm going to talk about how you can use the data that we annotate using sort of ontology as the structure. So first I'll talk about some kind of new thing we developed to represent the ontology and the annotated data using graph structure. And then some old tool that how you can peruse the ontologies using Worm Ontology browser. And then finally, we'll talk about how we use the annotated data, allow you to statistically analyze lists of genes. So in WormBase, we use a number of ontologies to annotate gene. So for function, we use gene ontology, molecular function, cellular component, and biological process. For gene expression, we use anatomy ontology, life stage ontology. So these are somewhat unique to the nematode. These ontologies we develop in-house, and also use gene ontology for subcellular localizations. For phenotype, we have phenotype ontology, again, somewhat nematode specific. And for human disease relevance for any given gene, we use the human disease ontology for the annotations. So SOBA graph, actually these will come online in the next release or two. We are still at the final stage of a little bit of a touch-up. And so there are basically 5 different graphs that you can find. You will be able to find them under the widgets, as Chris talked about, on the left side of every gene page. And so if you open up the expression widget, you will find the anatomy and life stage ontology SOBA graphs. SOBA stands for the summary of ontology-based annotation. And so in gene ontology, under the gene ontology tab, widget, human diseases, and phenotypes, respectively. So shown here is an example of the SOBA graph for LAT23, the LAT23 gene. So we go deeper into this part. So basically, the SOBA graph is taking all the annotations, in this case for LAT23, gene ontology annotations. We put them, overlay them on the ontology graph. And so the directly annotated nodes or terms are represented by the red circles, and indirectly or inferred annotations are the blue circles. And some special, terms with special importance. Then, in this case, the alliance, we call it alliance slip terms. You don't need to know that, but just so they are highlighted by with the shade. And you can manipulate this graph in some important ways. So, for example, you can restrict what kind of evidence type are used for the annotations. And you can narrow it to different aspects of gene ontology in this case. And you can also make this graph even simpler by restricting how many layers going from top to the leaf, how many layers you want to see. So the deeper you go, the more detailed the annotations will be shown. So here's an example of such restrictions. So LAT23 go graph again, but this time we apply the filter so that it's only showing experimental evidence annotations, and we limit that to a depth of 3. And so, and also this, I just want to also say that this graph is very interactive. You can click on each node and get more information. So for example, oops, sorry about that. And if you mouse over or click on any given node, I can't remember which one that was, you will see this information in terms of the specific counts of annotations and the term, which is hyperlink. You click on that, and you can go to the gene page. You can move these nodes around to get a better sort of a presentation. You can export the image for your presentation. So you should play with that when it becomes live. And so again, so you click on the node, you can go to this thing called the, well, you go to the ontology term page. On that page, there are a lot of information about that term. But specifically, we also have the ontology browser. And the ontology browser is, there are kind of three parts, but so you have a graph. You can look at this, but with respect to that term specifically, and you can look at it with this graph of the relationship that term has with other terms, and or in this kind of we call it inference tree view, because the ontologies are the terms are organized in hierarchy, and so that allows inferences. And this is an inference view. Again, you can look at the relationship, but very importantly, so again, on the ontology term page, in this WormBase ontology browser, that there's a count next to the focus term. There's a count of all the genes that have been annotated directly or indirectly with this term. You click on that, you get to a table or list of all the genes that are annotated with that term. And you can download them, and you can use them to do list operations, as Chris mentioned, for example, in WormBase. You can do other operations with this list of genes. You can download two different lists. So one, for example, phosphorylation in terms of gene ontology annotation, and neurons, say, for anatomy expression. So then, by combining those two gene lists, you can get at genes that are expressed in the neuron and involved in phosphorylation processes. And then another way to get at the ontology browser is through the front page, through the tools, and then ontology browser. And here is another way you can peruse the ontology, which is through this top-down kind of an interactive tree where you can, for any node, if it has children nodes, then, if you will, and you can click on the plus sign and then expands it, and you can gradually get to the leaf nodes. So this may be a good way for you to see what all the terms are there and find the terms you might be interested in using or getting more information on. Now, with all the information we have annotated using these ontologies, we can begin to offer a function that allow you to take your list of genes. So for example, from an RNA-seq experiment, a worm of a particular condition, you get through RNA-seq, you get a list of genes, and you wonder if there's anything special about this list of genes. And so this is a tool that basically you take the list and then sort of run it doing statistical analysis against all the genes we have for annotations, and to see if you see specific aspects in biology that appear within your list of genes. So here, again, from the tool, you can get to the enrichment analysis page where you paste a list of genes here, or you can upload a file with your list of gene names. And you can also filter out the background genes that always appear and you don't care about. And then you hit the analysis. And the result is that you get a table, a graph, and these are, they're pretty self-explanatory. But I just want to point out that all of these you can download. And also, you should pay attention to this section, which is that, in this case, I manually paste in E1 through E20 and ran the analysis, and it tells me that E19 doesn't actually exist, so it's invalid. It's a bad name, and there are 5 genes has no data relevant. In this case, it's a gene ontology. There's no gene ontology data for those genes. And only 14 genes are actually used in the analysis, and you should pay attention to that. So that's it. And the SOBA tool is, we have a test site, and you're welcome to come to our booth during the poster session and play with it, if we have good internet connection. Thank you.